Hey everyone, Dr. Steve here. You know it as well as I do, but most students need a B or better in A&P. But did you know that students are doing that all over the US using my Anatomy and Physiology Digital Suite as a low-cost alternative to expensive textbooks? Digital Suite delivers the key concepts of A&P with short, assignable tutor videos and a bulleted narrative study guide that not only explain the concepts of A&P, they show them happening. Combined with state-of-the-art dissection and physiology lab simulations and a vast question bank for quizzes, homework assignments, and exams, Digital Suite meets students where they are and gives them the tools to master the content. It's the perfect supplement to an OER textbook or just on its own. If you're an A&P instructor, contact your McGraw-Hill representative today and ask about Anatomy and Physiology Digital Suite. If you're a student, tell your instructor about it. There's a link to Digital Suite in the show notes of this episode. Get your B or better in A&P with A&P Digital Suite.
One day only Thanksgiving Day deals are coming to Lowe's.com. Plus, members get early access to online Black Friday doorbuster deals on gifting favorites, like the still trending Cobalt Mini Toolbox for just $14.98. Don't miss up to 50% off for one day only at Lowe's.com. We help, you save. Valid 11/27 only on Lowe's.com. Member only doorbusters and midnight Eastern. Loyalty program subject to terms and conditions. See Lowe's.com/terms for details. Subject to change while supplies last.
At Capella University, learning online doesn't mean learning alone. You'll get support from people who care about your success, like your enrollment specialist who gets to know you and the goals you'd like to achieve. You'll also get a designated academic coach who's with you throughout your entire program. Plus, career coaches are available to help you navigate your professional goals. A different future is closer than you think with Capella University. Learn more at capella.edu.
Hey everyone, welcome back to my podcast, Anatomy and Physiology Bit by Bit. I'm your host, Dr. Steve Sullivan, coming to you from Bucks County, PA, where I've been teaching A&P since 2002. Today's episode is about evolution and natural selection. Why is this important to anatomy and physiology? Because this is ultimately about the origin of all the anatomical structures and physiological processes we see in not only humans, but all species on the planet. Some may say this is a controversial topic, but it shouldn't be. There is no controversy among the biological community. Multiple surveys have revealed that about 99% of all biologists accept evolution and natural selection as a scientific certainty. So I won't treat evolution as a controversial subject. And when it comes to alternative theories for human origins, I would only entertain those that follow the scientific method. Evolution is, at its heart, a story about families. Very, very big families stretched across millions of years. It's the story of how traits are passed down, how small differences arise, and how those differences can, over long periods of time, reshape life on Earth. In this episode, we'll walk through what evolution is, how natural selection works, how heredity makes evolution possible, and how different patterns of evolution, like divergent and convergent evolution, shape the living world. Then we'll tackle some of the most common myths and misconceptions that often cause confusion. But speaking of the scientific method and scientific theories, I think I should start by explaining what a scientific theory is and what it isn't. Science is, at its core, a disciplined way of being curious. Imagine you notice something odd, like the fact that people in your class seem to come down with colds more often during exam week. You have a feeling that stress might be involved, but a feeling isn't enough. The scientific method is the process of turning that vague sense into a clear question, a testable explanation, and a careful investigation that anyone else could, in principle, repeat. It usually begins with an observation. You see a pattern. More sniffles and coughing in the library around midterms, or more students missing class right after a big exam. From that observation, you move to a question. Does stress actually increase the likelihood of getting sick? The question matters because it has to be specific and testable. Are exam weeks miserable isn't a scientific question. Does higher stress correlate with more colds? Is. From there, you form a hypothesis, a tentative explanation or prediction. You might say, If students are under more stress, then their immune function will decrease, and they will get sick more often. A good hypothesis is not just a guess, it's a claim that could be wrong, and clear evidence is possible to make it wrong. It must be falsifiable, meaning there is some possible result that would show it isn't true. If, after careful study, highly stressed students turn out to be sick less often, that would challenge your hypothesis. We often hear unfalsifiable claims, and that is typically unscientific. For example, someone makes a claim like, "Ghosts are making noise in my house." Someone else says, "Can I see the evidence of that?" And the response is, "Well, you can't see or hear them. They don't have a smell, etc." There's no way to actually prove that ghosts are in my house. That provides no possibility of proving them wrong, which makes the claim unfalsifiable. It cannot be falsified by any means. In this case, the claim is not scientific. It's supernatural, and therefore it cannot be tested with the scientific method. Next comes the step that makes a hypothesis concrete: prediction. You translate the hypothesis into specific expectations about what you'll see if it is correct. For instance, you might predict that students who report higher stress on a standardized survey will have more colds, more doctor visits, and more days absent due to illness during exam periods than students with lower stress scores. These predictions guide you in designing your study. To test those predictions, you design and carry out an experiment or an observational study. In this case, you might recruit a group of students, give them a validated stress questionnaire, and then track their health over a semester. You might record how many times they catch a cold, whether they visit the campus clinic, or how many days they miss class due to illness. You'd also try to account for other factors: sleep habits, smoking, underlying health conditions, because those can influence both stress and illness. In many experiments, scientists include control groups, so they can see if the incidences are not also occurring at the same rate in groups that don't have any stress. They also might use random assignment or blind participants and researchers, making sure that nobody knows the details of each group so there's no bias in their data collection. The details vary by field, but the aim is the same: to isolate the effect of the variable that you care about. Once you collect your data, you move on to analysis. This is where statistics come in. You're asking, do the results match what I expected if my hypothesis is true? Do highly stressed students really get sick more often, and is the difference big enough that it's unlikely to be just due to random chance? If the data line up with your predictions, you say that your results support your hypothesis. If they don't, you say your hypothesis is not supported and may need to be revised or rejected. Notice that in science, even a well-supported hypothesis is not declared proven in an absolute sense. It is simply the best supported explanation we have so far. After you've analyzed the data, you draw conclusions, but the process doesn't end there. Scientists share their methods and results, usually in the form of research papers. Other experts scrutinize the work through peer review, asking whether the methods were sound, the analysis was appropriate, and the conclusions were justified. Other groups may then try to replicate the experiment, repeating it or testing the hypothesis in a slightly different way. When independent researchers repeatedly find similar results, confidence in the explanation grows. When they don't, that's a signal that something in the original hypothesis, the methods, or the assumptions may need to be re-examined. This looping process, observation, question, hypothesis, prediction, test, analysis, revision, and retest, is what we call the scientific method. It isn't a rigid checklist. Different fields and different questions may tweak the order or style, but the core spirit is consistent. Science is about building explanations that are anchored in evidence, always open to being refined or replaced if new, better evidence appears. Within this framework, it's essential to understand what scientists mean by facts and theories because those words often cause confusion. A scientific fact is, in simple terms, a repeatable observation. It's something that has been measured or seen consistently enough that we are very confident it is true in the conditions described. For example, objects dropped near Earth's surface accelerate downward at about 9.8 meters per second squared, if you're ignoring air resistance. Water at sea level boils at about 100 degrees Celsius. In medical research, it's a fact that in large studies, smoking is associated with higher rates of lung cancer. These are all statements about what happens when we look at the world in a particular way. Facts, however, are pieces of information. They tell us that something occurs, but not necessarily why it occurs. Observation that bacteria are often present in infected wounds is a fact. Explaining how those bacteria cause disease takes us to a different level of scientific understanding. That's where scientific theories come in. In everyday language, people often use theory to mean a guess or a speculation, an opinion. Someone will say, I have a theory about why the traffic is so bad today. In science, the word theory means almost the opposite. A scientific theory is a well-substantiated explanation of some aspect of the natural world, built from a large body of evidence and capable of making testable predictions. The theory of evolution by natural selection, the germ theory of disease, the cell theory, the theory of plate tectonics, and atomic theory are all examples. Take germ theory. For centuries, people noticed facts. Wounds sometimes got infected. People exposed to sick individuals often became sick themselves. Certain diseases seem to spread in patterns. Germ theory ties these facts together by proposing that many diseases are caused by microorganisms, bacteria, viruses, fungi, and those microorganisms invade the body and interfere with its normal function. This theory not only explains why bacteria appear in infected wounds, it also predicts that sterilizing surgical instruments will reduce infections, that antibiotics targeting certain bacteria will cure specific diseases, and that vaccines that train the immune system against a pathogen can prevent illness. Those predictions have been tested again and again, and overwhelmingly are supported by evidence. A similar story holds for other major theories. Evolutionary theory, that we're going to talk about later today, explains why we see nested patterns of similarity in DNA and anatomy, why certain fossils appear in the order they do, and why organisms are distributed as they are across the planet. Plate tectonic theory explains the matching shapes of continents, the distribution of earthquakes and volcanoes, and the formation of mountain ranges. Atomic theory explains chemical reactions, states of matter, and the structure of the periodic table. These theories are not guesses waiting to become facts. They are sophisticated frameworks that organize huge numbers of facts and smaller, more specific hypotheses into coherent, testable explanations. So how do facts and theories relate? It's helpful to stop imagining them as rungs on a ladder where ideas start as guesses, become hypotheses, then theories, and then finally graduate into facts. That is not what is happening here. Instead, think of them as different kinds of knowledge that work together. Facts are like the individual bricks, specific observations and measurements. Theories are like blueprints or architectural plans. They show how the bricks fit together into a structure, and they tell you what new rooms you could build and what would happen if you added or removed a wall. There is another term people often hear, and that is scientific law. A law is usually a concise statement, often mathematical, that describes a consistent relationship in nature. Newton's law of gravitation, Ohm's law for electrical circuits, or the ideal gas law are examples. They tell you what will happen under certain conditions, how force relates to mass and acceleration, how voltage relates to current and resistance, or how pressure, volume, and temperature of a gas interrelate. Like facts, laws don't usually explain why. That's the role of theories. General relativity, for instance, explains gravity in terms of the curvature of space-time and helps make sense of where Newton's law works well and where it needs correction. Don't think of a theory as graduating to a law. That is not how it works. All of this matters because misunderstandings about terms can spill over into misunderstandings about the reliability of science. When someone says evolution is just a theory, they may be using theory in the everyday sense. But in scientific language, calling evolution a theory is saying that it is a deeply supported, explanatory framework grounded in countless facts from genetics, paleontology, comparative anatomy, biogeography, and even more. The same goes for saying germs are just a theory, or atoms are just a theory. technically true in the scientific sense, but completely backwards if you interpret theory as a mere guess. The scientific method does not promise absolute, unchangeable truth. Instead, it offers something more practical and powerful, a way to build explanations that are reliable, testable, and open to revision. No matter how well supported, any theory can, in principle, be modified if new, solid evidence demands it. But that doesn't mean everything is perpetually up for grabs. Some theories are so well tested that overturning them would require not just new data, but also a new explanation that can account for everything the old theory explained and more. In everyday life, this process is what helps us trust scientific conclusions. When multiple independent lines of evidence converge on the same explanation, when experiments are repeated and yield similar results, when new observations from new technologies, like better telescopes or DNA sequencing, keep fitting into the existing framework and refining it rather than demolishing it, that's how we know we're getting closer to how the world actually works. So the scientific method is not just a school diagram. It is a living process. We observe, we ask, we propose, we test, we revise. Along the way, we accumulate facts, we build and refine theories, and we occasionally discover new laws that capture patterns in concise form. Together, those pieces give us a remarkably effective way to understand and navigate the natural world. Okay, so, scientific method. You get it. Science is important. So now, let's talk about evolution. When biologists talk about evolution, they mean a change in the genetic makeup of a population over generations. It's not about an individual animal deciding to evolve. It's about small shifts in which traits are more common in a group of organisms over time. This is not controversial at all. We can all observe this happening. Think about antibiotic-resistant bacteria. These are bacteria that have evolved a resistance to antibiotics we've been using against it. Genetic change over time in a population is irrefutable. There are three key ideas at the core of evolution. Variation, individuals in a population are not identical. Heredity, some of these differences are passed down from parents to offspring. And differential success, some traits help organisms survive or reproduce better in their environment. Put those together over many generations, you get evolution. You might think of a population, say like a group of rabbits, as a kind of cloud of genetic possibilities. Within that cloud are different fur colors, ear lengths, speeds, immune system strengths, and so on. When the environment filters which traits work best, the genetic cloud slowly shifts. That shifting of the cloud, that's evolution. Okay, for evolution to happen at all, there has to be heredity, a way for traits to be passed from one generation to the next. Having said that, if you haven't listened to episode 79 on genetics and heredity, you may want to pause this, go listen to episode 79, and then come back. Understanding genetics and heredity is very helpful to understanding evolution. I'll give a quick summary of DNA, genes, and traits, and then we'll move on. Inside almost every cell of your body is DNA, arranged into chromosomes. Segments of DNA that carry instructions for making proteins or regulating biological processes are called genes. Those genes influence traits, eye color, blood type, lactose tolerance, beak shape, and so on. Each gene can come in different versions called alleles. For example, the gene that influences human ABO blood type comes in alleles A, B, and O. The combination of alleles you inherit from your parents helps determine your traits. Two key points here. One, you don't inherit traits directly. You inherit genes. So you don't inherit being tall. You inherit many genes that influence growth, bone structure, hormone levels, etc. The final result, your actual height. That comes from those genes interacting with nutrition, health, and other environmental factors. Two, populations carry a pool of alleles. The genetic variation in the population acts as the raw material for evolution. Natural selection works on that variation. So where does the variation come from? Well, it comes from several sources. First, you have mutations. Random changes in DNA. They can occur when DNA is copied or due to radiation, chemicals, or spontaneous chemical changes. Most mutations are neutral. Some are harmful, a few are helpful in a given environment. Recombination. Remember from my episode that covered meiosis and spermatogenesis and oogenesis, during the formation of eggs and sperm, chromosomes from each parent are shuffled and swapped. This recombines alleles into new combinations given to the offspring. Sexual reproduction is another one. When two parents combine their genetic material, each offspring gets a unique mix of alleles. These three processes constantly introduce and reshuffle variation, giving evolution something to work with. One of the most powerful pieces of evidence for evolution is common heredity, the idea that all life on Earth shares a common ancestor way back in the past. The genetic code is nearly universal. A codon that means start, or methionine, in a bacterium basically means the same thing in a human cell. Many genes are shared among very different organisms. You have genes related to cell division, DNA repair, and basic metabolism that are recognizably similar to those in yeast, plants, and bacteria. Even complex structures like eyes, limbs, and nervous systems are built using related gene families from one organism to the next. This shared genetic toolkit suggests that life didn't start separately many times over with completely different designs. Instead, life diversified from earlier shared ancestors with genes carried forward and then modified over time.
Get ready to shop Lowe's Black Friday doorbuster deals to save $150 on a 30-inch Blackstone griddle, now $299. Plus, choose a Whirlpool top-load washer or Midea top freezer refrigerator for just $398. These limited-time doorbuster deals go fast, so get holiday ready today at Lowe's. We help, you save. Valid 11/28 through 12/1. Selection varies by location. While supplies last. See Lowe's.com for more details.
At Capella University, learning online doesn't mean learning alone. You'll get support from people who care about your success, like your enrollment specialist who gets to know you and the goals you'd like to achieve. You'll also get a designated academic coach who's with you throughout your entire program. Plus, career coaches are available to help you navigate your professional goals. A different future is closer than you think with Capella University. Learn more at capella.edu.
So now we need to talk about the central mechanism of evolution. Like, how does evolution work? What causes it? And that is natural selection. In the mid-1800s, Charles Darwin and Alfred Russel Wallace both realized something profound. If organisms differ, and those differences affect survival and reproduction, then nature itself can act like a selective breeder. Over time, traits that help organisms survive and reproduce become more common. That's natural selection. We can break it down into 4 conditions that are required for natural selection. 1 would be variation. Individuals in a population differ in their traits. 2, inheritance. Some of these differences are heritable. They're genetic. You can pass them on to offspring. Overproduction. More offspring are produced than can survive and reproduce. And differential survival and reproduction. This is fitness. Individuals with certain traits are more likely to survive in their environment and produce more offspring. Remember, when we talk about fitness as an evolutionary term, we're not talking about being big, strong, and fast. We're talking about how well you fit in the environment. That's the fitness they're talking about. When these conditions are met, these 4 conditions, variation, inheritance, overproduction, and differential survival and reproduction, the proportion of beneficial traits increases over generations. Here's a concrete example: rabbits and their predators. Imagine a population of wild rabbits living in a field where coyotes hunt. 1, we have variation. Some rabbits are slightly faster, some have better hearing, some are better camouflaged. We have inheritance. Faster rabbits tend to have faster offspring. Camouflaged rabbits tend to have more camouflaged offspring. And rabbits with better hearing tend to have baby rabbits with better hearing. This is inheritance. We also have overproduction. Each rabbit pair can produce many offspring. but not all of them survive. This means that enough offspring are being created that if some don't make it, the population still exists. And then we have differential success. Coyotes catch more slow rabbits, while faster rabbits escape more often and live long enough to reproduce. Then there's differential success. Faster rabbits can outrun the coyotes, not get eaten, live long, reproduce. Rabbits that can hear the coyotes coming better can avoid being caught. Rabbits that are camouflaged are harder for the coyotes to find. All of those traits increase the potential survival of those individuals, increase their ability to reproduce, and increase how many new offspring will share those traits. So after many generations, the average speed in the rabbit population increases. The number of camouflaged rabbits increases. The number of rabbits with better hearing increases. No one rabbit tried to be faster, tried to hide better, or tried to hear better. The environment, meaning the existence of coyotes and their desire to eat rabbits, favored the fast rabbits, the better hearing rabbits, and the better hidden rabbits. So their genes become more common because they're the ones who are giving their genes off to offspring. That is natural selection in action. Let's look at some real-world traits and how natural selection has shaped them. I mentioned antibiotic resistance in bacteria. Bacteria in your body or in a hospital setting vary in their susceptibility to the antibiotics that we produce. Before exposure to an antibiotic, some bacteria happen to carry mutations that make them slightly less affected by that drug. When the antibiotics are applied, most bacteria die, but those with resistance survive. Those survivors multiply, pass their genes on to their offspring, leading to a population dominated by resistant bacteria. The trait here is antibiotic resistance, a genetically based difference in how well bacteria can survive in the presence of a drug. Selection has favored the resistant bacteria because they left more descendants in that environment. Let's talk about lactase in humans, the enzyme that breaks down milk sugar or lactose. All human infants can digest lactose, which is the sugar in milk, because they produce an enzyme called lactase. In most mammals, lactase production declines after we stop nursing. But in some human populations, adults continue to produce lactase and can digest milk as adults. This is called lactase persistence. In populations that domesticated cattle or other milk-producing animals, being able to digest milk in adulthood meant access to a rich food source. Individuals who could digest lactose had a nutritional advantage, especially in times of food scarcity. So over generations, alleles for lactase persistence became more common in those populations. This is a clear example of a trait, adult milk digestion, shaped by natural selection in specific human environments. Another human example is one I covered in my genetics episode, the sickle cell trait. A mutation in the gene for hemoglobin, the oxygen-carrying protein in red blood cells, can cause red blood cells to assume a sickle shape under low oxygen. Having 2 copies of this mutation can cause sickle cell disease, which is serious and life-shortening. But individuals with 1 copy have an interesting advantage in regions where malaria is common. The sickle cell trait makes it harder for the malaria parasite to complete its life cycle in the red blood cells. As a result, carriers of the sickle cell trait are less likely to die from malaria. In areas with intense malaria, natural selection favors the heterozygous state. Enough normal hemoglobin to avoid severe disease most of the time, plus enough altered hemoglobin to protect against malaria. This is why the sickle cell allele is relatively common in certain populations. Okay, let's talk about something called divergent evolution. This occurs when 2 or more descendant species evolve different traits from a common ancestor, often because they are adapting to different environments or lifestyles. The key idea is common origin, diverging outcomes. Biologists often point to homologous structures as evidence of divergent evolution. These are structures that share a common underlying anatomy because they come from a shared ancestor, even if they now serve very different functions. A classic example is the forelimbs of vertebrates. A human arm, a bat's wing, a whale's flipper, a dog's front leg. These are all homologous structures. They have the same basic pattern of bones. One upper bone, in the human it's humerus, two forearm bones, the radius and ulna, many wrist bones, the carpals, and a set of many digits. Yet the functions of these limbs are very different. In humans, we're grasping. With bats, we're flying. With whales, we're swimming. With dogs, we're running. The best explanation is that these species inherited a basic limb structure from a common ancestor, and then, over millions of years, natural selection modified that structure for different tasks. That's divergent evolution. Sometimes a single ancestral species gives rise to many descendant species that occupy a wide range of ecological roles. This is called adaptive radiation, a special case of divergent evolution. Consider Darwin's finches in the Galapagos Islands. The ancestral finch species likely arrived from mainland South America. On different islands with different food sources, seeds, insects, cactus nectar, different traits were favored. Over time, different species evolved with beaks specialized for cracking tough seeds, probing for insects, or sipping nectar. All of these finches share a common ancestor but have diverged into many forms, each adapted to a different niche. Now, if we have divergent evolution, then we also have convergent evolution. Different origins, similar solutions. So this is in contrast to divergent evolution. It happens when unrelated lineages independently evolve similar traits because they face similar environmental challenges or opportunities. Here, the idea is different origins, similar outcomes. Now we talk about analogous structures. Convergent evolution often produces these analogous structures, which are features that serve similar functions and may look similar, but do not come from a shared ancestral structure. A classic example, the wings of birds, bats, and insects. Bird wings are modified forelimbs with feathers. Bat wings are also modified forelimbs with skin stretched between elongated fingers. Insect wings are completely different structures growing from the exoskeleton. These wings are analogous, not homologous. They evolved independently in response to the challenge of powered flight. All of these organisms had an advantage by being able to fly. That advantage arose independently of a common trait because the environments that they lived in required it. Another striking example is the streamlined body shape of dolphins, sharks, and extinct marine reptiles like ichthyosaurs. Sharks are cartilaginous fish. Dolphins are mammals, more closely related to land mammals like cows and hippos than they are to fish. Ichthyosaurs were reptiles, more closely related to lizards. Despite these very different ancestries, all three groups evolved torpedo-shaped bodies, fins or flippers, narrow tails for propulsion. They evolved similar traits because they all lived in the same environment, even though their common ancestor was way back in the past, so those features evolved separately. The ocean rewards a streamlined shape that reduces drag and allows for swift swimming. Natural selection has repeatedly discovered this solution in unrelated lineages. Another example is desert plants, like a cactus and euphorbs. In North and South America, you find cacti with thick, water-storing stems and spines instead of leaves. In parts of Africa, you find euphorbs that look remarkably similar, thick, fleshy stems and spines, but they belong to a completely different plant family. These similarities evolved independently as adaptations to hot, dry environments. Again, different lineages converged on similar solutions, illustrating convergent evolution. So natural selection is central, but it's not the only way evolution happens. A few other forces matter too. We have genetic drift, random changes in allele frequencies, especially in small populations. For example, if a storm wipes out most of a small population at random, the surviving alleles might not be the best ones, just the ones that happen to be there. We have gene flow. movement of alleles between populations due to migration. If members of one population move into another and reproduce, they introduce new alleles. And sexual selection, a form of natural selection where the key factor is success in mating. Traits that attract mates, even if they're costly or risky, can become more common. Think of the elaborate tail of a peacock, or complex bird songs. These processes all contribute to how populations change over time. If we have enough time, and we have, differences can accumulate to the point where populations become new, distinct species. A species is often defined in sexually reproducing organisms as a group whose members can interbreed and produce fertile offspring under natural conditions. Speciation, which is the transition from one species to another, can happen through several routes, such as geographic isolation. A population gets split by a barrier, like a new river, a mountain range, or an island. Each population experiences its own mutations, selection pressures, and drift. Over time, they diverge genetically and behaviorally. Eventually, even the barrier disappears, but they may no longer be able to interbreed successfully. Another one is ecological or behavioral isolation. Even without a physical barrier, populations might specialize on different habitats or develop different mating behaviors, leading to reduced interbreeding and eventual speciation. When enough differences accumulate, the reproductive isolation is complete. Biologists recognize two different species. This is evolution on a larger scale, often called macroevolution, but it's driven by the same fundamental processes we see at smaller scales. Okay, the last thing I want to talk about are the myths and misconceptions about evolution. Evolution is sometimes misunderstood, and these misunderstandings can make the whole subject seem confusing or controversial. Let's clear up some of the most common misconceptions. 1. I talked about this before. Evolution is just a theory. I think it's still worth mentioning as misconception number 1, but I won't reiterate it. We can move on to number 2. This is probably the biggest error people make when they're arguing against evolution. If they say evolution is just a theory, then what I hear is you don't really understand how science works. Number 2, individuals evolve. You do not personally evolve in the evolutionary sense. You can grow, learn, and adapt behaviorally, but evolution happens at the population level across generations. Your DNA sequence is largely fixed from the moment of conception. Evolution occurs when the frequency of alleles changes in a population from one generation to the next. So a person doesn't really evolve from a natural selection perspective. Number 3, evolution says we came from monkeys. This is false. Evolution does not say we came from monkeys. Evolutionary biology does not claim that humans came from modern monkeys like chimpanzees. Instead, it says that humans and other primates share common ancestors. Think of it like this. You and your cousins share grandparents. You didn't come from your cousin. You both came from earlier family members. Similarly, humans and chimpanzees share a common ancestor that lived several million years ago. From that ancestral population, different lineages split and evolved along different paths. One lineage eventually led to modern humans, another led to modern chimpanzees. So it's more accurate to say humans and other primates share common ancestry. Another misconception is people say evolution has goals or a direction. They'll say, why aren't chimpanzees humans by now? It's tempting to talk about evolution as if it were trying to produce more complex or better organisms, but that's misleading. Natural selection has no foresight, no plan, no goal. It simply favors traits that are beneficial here and now in a given environment. If being smaller and simpler has advantages, evolution can favor that. Many parasites are simpler than their ancestors. If being more complex helps in a particular setting, then complexity may increase. But there's no cosmic ladder with humans on top. Evolution is more like a branching tree than a straight line, with countless lineages adapting to their own circumstances. Another misconception is that evolution is entirely random. I've heard people say, I don't think that humans could have arisen randomly. Well, there is randomness in evolution. DNA mutations occur without regard to whether they're needed. and some changes are due to chance events like genetic drift. But natural selection itself is not random. Those random mutations introduce variation. Non-random selection filters that variation. Some traits consistently do better than others in a given environment. It's a bit like shuffling a deck of cards. That's random. And then playing a game with rules that reward certain combinations. That's not random. The shuffling is random. The outcomes favored by the rules you've made are not. Number 6. There are no transitional fossils. This is the missing link question. People say, well, where's the missing link? This is a misunderstanding of what evolution actually is. A transitional fossil is one that shows a combination of traits from ancestral and descendant groups. There are many such fossils. Fossils that show features between land-dwelling mammals and fully aquatic whales. Fossils that show feathered dinosaurs with a mix of bird-like and reptile-like features. Fossils that document the gradual changes in horse limb bones and teeth over tens of millions of years. We don't have a fossil of every individual that ever lived. That would be impossible. But we have enough transitional forms to clearly show patterns of gradual change and branching lineages.
At Lowe's, before the holidays is the perfect time for upgrades and upkeep. During Black Friday, get up to 50% off select major appliances and buy more to save more with up to an additional 25% off when you bundle select major appliances. Plus, grab select DeWalt 20-volt max drill or impact driver kits for just $99. Lowe's. We help, you save. Valid through 12/3. Selection varies by location. While supplies last. See Lowe's.com for more details.
At Capella University, learning online doesn't mean learning alone. You'll get support from people who care about your success, like your enrollment specialist who gets to know you and the goals you'd like to achieve. You'll also get a designated academic coach who's with you throughout your entire program. Plus, career coaches are available to help you navigate your professional goals. A different future is closer than you think with Capella University. Learn more at capella.edu.
Number 7. Accepting evolution means rejecting all religious or spiritual beliefs. This is more of a philosophical and personal issue than a scientific one, but it's important to recognize that people hold a wide range of views. The science of evolution describes how life changes over time and explains patterns we observe in genetics, anatomy, and the fossil record. It does not and cannot pronounce on questions like ultimate purpose, meaning, or the existence of the divine. Those are philosophical or theological questions, not empirical ones. They can't be tested with the scientific method. Many individuals and religious traditions find ways to reconcile evolutionary science with their beliefs, viewing evolution as the natural process through which life's diversity unfolded. Others interpret their traditions differently. From a scientific standpoint, the key point is that evolution is about how life changes, not about why there is life at all in any deeper metaphysical sense. The late Stephen Jay Gould, a renowned Harvard University paleontologist and evolutionary biologist, called religion and science non-overlapping magisteria, abbreviated as NOMA. It means science deals with the empirical world of facts and theories, while religion addresses questions of moral meaning, values, and ultimate purpose. NOMA aimed to show how these two fields can coexist without conflict by respecting the boundaries of each magisterium or domain of teaching authority. In fact, long before Gould, the famous scientist Galileo Galilei was quoted as saying, "The Bible shows us how to go to heaven, not how the heavens go." Basically the same thing. Now, Galileo lived in modern-day Italy, not far from the Vatican, in the 15 and 1600s. In fact, the Vatican under Pope Urban VIII sentenced Galileo to house arrest for the last 9 years of his life because in his book Dialogue, he seemed to defend the Copernican theory that the sun was the center of the solar system, despite Bible passages implying that the Earth is the center of the solar system. So he was found vehemently suspect of heresy. While his perspective was the Bible, you can substitute any religion in his quote, and it still applies. One more thing I'd like to mention about common criticism of evolution. Often when I'm talking about evolution with someone who holds an opposing view, they bring up the Big Bang, how the universe was created, or they bring up how life began on Earth. The problem is that evolutionary theory and natural selection do not address how the universe was created, nor how life began on Earth. It simply explains how we have the diversity of life on Earth that we can see today and in the fossil record. How life began, we do not know with any certainty. Criticizing a theory for not explaining something it doesn't claim to explain is an unfair criticism. It's akin to a straw man argument. This is a debate tactic in which someone chooses to avoid addressing the actual topic and instead creates a position for their opponent they can easily argue against and win. It's like challenging someone to a fight, and instead of fighting the actual person, you build a straw man in their likeness and fight that instead. The last misconception I hear a lot is that not enough time has passed for evolution to occur at the degree we see it. Well, one thing we didn't address much is time. In most cases, the accumulation of changes in inherited traits that are selected by nature takes a very long time to appear in a population's phenotype. Ultimately, this is when we see speciation and when we see extinction. The individuals who had the traits that could not overcome the selection pressure eventually die off. When we look back at the fossil record, we see that as different than any species that persisted. That's an extinct species. Speciation can take thousands, if not more, generations to occur, so we need a lot of time. The good news is that we've had a lot of time. The universe was created about 14 billion years ago, and the Earth was formed about 4.5 billion years ago. Life began on Earth about 3.5 billion years ago. But how do we know this? Well, before the 20th century, people tried to estimate Earth's age in all sorts of ways. Some geologists looked at how fast rivers deposit sediment and extrapolated backward. Others, like Lord Kelvin, used physics. He treated Earth like a hot iron ball cooling in space and came up with tens of millions of years. Those numbers were all over the place, and they all had a fatal flaw. They assumed Earth was just passively cooling and didn't know about radioactive heat or the constant recycling of rock. The real breakthrough came with something no one had anticipated: radioactivity. In the early 1900s, scientists discovered that some atoms are unstable. A uranium atom, for example, will eventually decay into lead, changing its identity in a predictable, statistical way and emitting radiation along the journey. We'll refer to the original element as the parent, and the product of its decay the daughter. Each radioactive isotope has a characteristic half-life, the time it takes for half of a given amount to decay into its daughter product. That gave geologists a remarkable kind of clock. When a rock mineral crystallizes from molten material, it locks in a certain number of parent atoms, like uranium, and essentially 0 daughter atoms, like lead, because the daughters are excluded from the crystal structure. As time passes, the parent decays into the daughter at a known rate. By measuring the ratio of parent to daughter atoms today, and knowing the half-life from laboratory physics, you can calculate how long that decay has been going on. This method is called radiometric dating, and it's not just one technique. There are many parent-daughter pairs: uranium-lead, rubidium-strontium, potassium-argon, and others, each with its own half-life and strengths. When different methods on the same sample give the same age, our confidence that we're reading a genuine timestamp goes way up. You might think, just date the oldest rocks you can find on Earth, and that's the planet's age. Unfortunately, Earth is geologically restless. Plate tectonics constantly recycles crust, melting old rock, forming new rock, and metamorphosing existing rock. Erosion and burial shuffle materials around, and heating can cause daughter atoms like argon gas to leak out, resetting some isotopic clocks. So the oldest rocks we still have intact are younger than the Earth itself. The oldest known rock formations are a little over 4 billion years old, and individual zircon crystals inside younger rocks have been dated to about 4.4 billion years. Those zircons tell us Earth's crust existed by then, but they still don't pin down the exact birth date of the planet. To get the age of the Earth, scientists stepped back and asked, when did the whole solar system form? Earth, after all, formed along with other planets from the same swirling disk of gas and dust. Here's where meteorites come in. Many meteors are chunks of primitive material that solidified very early in solar system history and have remained chemically closed ever since. When a meteor enters the Earth's atmosphere and lands on its surface, it becomes a meteorite. When we apply radiometric dating to these meteorites, especially using very robust methods like uranium-lead and lead-lead dating, we consistently get ages around 4.54 and 4.57 billion years for the oldest solid bits, which are called calcium-aluminum-rich inclusions. And Claire Patterson famously pulled all this together in the 1950s, analyzing isotopes in meteorites and arriving at an age of about 4.55 billion years. That value has been refined, but not overturned. We also date moon rocks, which cluster around 4.5 billion years old, and meteorites from Mars, which also fall near this age range. Everything points to a coherent picture. The Sun and its planets formed roughly 4.55 billion years ago, and Earth reached essentially its present mass around the same time. So the age of Earth is not a single measurement from a single rock. It's the convergence of meteorite ages, lunar samples, the oldest terrestrial minerals, and models of how planetary systems form. All of them tell the same story: an approximately 4.5 billion year old planet. So the next question is, when did life begin on this planet we have? Once Earth existed, it had to cool enough to form a solid crust with liquid water on its surface before life could plausibly arise. Those ancient zircon crystals from Western Australia don't just tell us about age. They also carry isotopic clues about the conditions they formed in. Some zircons, dated around 4.3 billion years ago, contain oxygen isotope signatures that are best explained by interaction with liquid water at the surface. That implies that the Earth had oceans, and therefore a relatively cool crust and atmosphere. And that was within a few hundred million years of forming. So by about 4.3 billion years ago, the Earth had a crust, it likely had stable bodies of water, and the environment was at least physically capable of hosting life. The big question becomes, when did life actually start? We will probably never find the first cell as a fossil, but we do have multiple lines of evidence that push the origin of life back very far into the past. The most straightforward evidence comes from structures and microfossils that preserve the remains or handiwork of ancient microbes. One key player: stromatolites. Stromatolites are layered, mound-like structures built by microbial communities, often dominated by photosynthetic cyanobacteria. Microbes form slimy mats that trap sediment over time. Layers pile up into a laminated rock structure. In Western Australia's Dresser Formation and other ancient rock units, geologists have found stromatolite fossils that are about 3.48 to 3.5 billion years old. These structures are so strikingly similar to younger, clearly biological stromatolites that they are widely regarded as direct evidence of living microbial communities at that time. Other sites, such as slightly younger rocks in South Africa and additional localities in Australia, also preserve microfossils and stromatolite-like features from around 3.4 to 3.5 billion years ago, again pointing to a thriving microbial biosphere by then. These rocks are dated using the same radiometric methods described earlier, applied to volcanic layers or minerals in the same sequence. The fossils, in turn, are dated by their position within those dated rocks. From just this line of evidence, we can say with confidence that by 3.5 billion years ago, Earth had extensive microbial life, including organisms capable of building stromatolites. But biology leaves more subtle traces than visible fossils do. Living organisms tend to prefer certain isotopes. For example, they use an isotope of carbon called carbon-12, slightly more readily than carbon-13 when building organic molecules. This preference shows up as a distinctive isotopic signature in carbon-rich material. Scientists can measure the ratio of carbon-12 to carbon-13 in ancient rocks. If the ratio is unusually enriched in carbon-12, and if there is no obvious non-biological process that would create that pattern, then it's reasonable to interpret that carbon as biogenic, meaning the remains of ancient life. Using these methods, researchers have found carbon with a biological-looking isotope in 3.7 billion-year-old rocks from Greenland. Even more striking, graphite inclusions, which are carbon, were found inside a 4.1 billion-year-old zircon grain from Australia whose isotopic composition is also compatible with life. Because that graphite is sealed inside a very old crack-free zircon crystal, it's extremely unlikely to be contamination. That doesn't absolutely prove it's from life. Some non-biological processes might mimic that signature, but it's a strong hint that life might already have existed by 4.1 billion years ago. These older claims are more controversial than the 3.5 billion-year-old stromatolites, and scientists debate the details, but they push the plausible origin of life very close to the time when oceans first formed. Okay, let's pull all of this evidence together. Earth forms about 4.54 billion years ago. Stable oceans likely appear by about 4.3 billion years ago. And clear, visible fossils, stromatolites and microfossils, are widespread by 3.48 to 3.5 billion years ago. From this convergence, most scientists are comfortable saying life on Earth has certainly existed for at least 3.5 billion years, and it may have originated quite a bit earlier, possibly as far back as 4.1 billion years ago, not long after the Earth became cool and wet enough to be habitable. We do not yet know the exact date when the first true cell assembled, and we may never pin that down to a precise number. The earliest steps probably involved fragile molecules and structures that left little or no record in the rocks. But we can bracket the problem. By 3.5 million years ago, life is definitely up and running. By 4.1 billion, there may already have been life leaving its subtle isotopic fingerprints. So yes, there has been plenty of time to get the diversity we see on the planet by way of natural selection. Plenty of time. 3.5 billion years is a very, very, very long time.
One day only Thanksgiving Day deals are coming to Lowe's.com. Plus, members get early access to online Black Friday doorbuster deals on gifting favorites, like the still trending Cobalt Mini Toolbox for just $14.98. Don't miss up to 50% off for one day only at Lowe's.com. We help, you save. Valid 11/27 only on Lowe's.com. Member only doorbusters and midnight Eastern. Loyalty program subject to terms and conditions. See Lowe's.com/terms for details. Subject to change while supplies last.
At Capella University, learning online doesn't mean learning alone. You'll get support from people who care about your success, like your enrollment specialist who gets to know you and the goals you'd like to achieve. You'll also get a designated academic coach who's with you throughout your entire program. Plus, career coaches are available to help you navigate your professional goals. A different future is closer than you think with Capella University. Learn more at capella.edu.
Okay, why does evolution and natural selection matter? Well, understanding these things isn't just an abstract exercise, it has practical consequences. For example, medicine. We manage antibiotic resistance, design vaccines, and study rapidly evolving pathogens like influenza and coronaviruses using evolutionary principles. Agriculture. Crop breeding, pest control, and livestock management rely on understanding how traits are inherited and how populations respond to selection. Conservation biology. Protecting endangered species and maintaining genetic diversity requires understanding how small populations evolve and how they may or may not adapt to changing environments. And human health. Traits like lactose tolerance, susceptibility to certain diseases, and even aspects of our metabolism and immune system make sense in light of our evolutionary history. Evolution and natural selection give us a framework for understanding why organisms are the way they are, how they got that way, and how they might change in the future. Think about breeding dogs. Breeding dogs so that you get small little Chihuahuas or big Great Danes is all based on the principles of natural selection. So let's pull it all together. Life on Earth shares a common heritage encoded in DNA. That common heredity is why you can look at a human cell and a plant cell and recognize similar machinery at work. Over billions of years, mutations, recombination, and other sources of variation have constantly introduced new genetic possibilities. Natural selection acting on those inherited differences has shaped which traits become more common and which fade away. In one lineage, that meant fins becoming limbs and vertebrates walking onto land. In others, it meant limbs returning to the sea as flippers, or feathers evolving first for insulation or display and later becoming part of the machinery of flight. Sometimes lineages diverged, using a shared ancestral structure for wildly different purposes. That's divergent evolution, visible in homologous structures like the vertebrate forelimb. Other times, distant lineages converged on strikingly similar solutions to similar problems. That's convergent evolution, reminding us that physics, chemistry, and environmental constraints channel evolution's possibilities. Along the way, humans evolved too, shaped by nutrition, disease, social structures, and environments. Our capacity for language, culture, and technology adds new layers to how our species interacts with the world. But our bodies, our genes, and even many of our diseases still bear the marks of our evolutionary past. Evolution and natural selection don't diminish the wonder of life, they deepen it. Instead of seeing the living world as a static collection of unrelated forms, we see it as a vast, branching family tree, a dynamic history of change, adaptation, and survival written in DNA and recorded in fossils, anatomy, and behavior. And that story is still unfolding. Each generation adds a new chapter as populations continue to change, as environments shift, and as new traits emerge. To understand evolution is to read that story with clear eyes, but also to recognize that in a very real sense, we are part of that story. Okay, I get a little worked up when I talk about evolution, as you probably just heard. But it's really important to understand this scientific certainty when it comes to anatomy and physiology. So I hope you enjoyed that. I hope you learned something. If you have any questions, please feel free to email me at minus55media@gmail.com. I'll be happy to answer your questions. I hope this podcast is helping you get your B or better in A&P. Good luck, and I'll talk to you next time. Anatomy and Physiology Bit by Bit is a production of Minus 55 Media and is hosted by the Believe Network. Please take the time to rate my show and leave a review. You can also check out the content I post on Instagram and YouTube as @StudentHelp4AP. That's the number 4 AP. Special thanks to my family, Bucks County Community College, and McGraw-Hill Education, where your instructor can find Anatomy and Physiology Digital Suite, my low-cost tutor video-based digital learning solution for anatomy and physiology currently being used at several colleges and universities. If your instructor isn't using Digital Suite, let them know about it. The link is in the show notes.